Hello everyone. We discuss about the application of animal cell culture in last lecture. Now we are study the application of cell culture in the expression on the study of gene expression. That study we discuss about organ culture, explant culture and organotypic culture, all this. See this. What is the organ culture? Organ culture, it is a organ, whole organ that contain the cell. That cell, they cut the organ. That organ, we take one cell, and that cell grow in media. We culture whole organ, also called as organ culture. We discuss about that detail in next slide, but you otherwise you understand this. Then explant culture. Explant culture also it's a solid liquid interference. See this. This is a solid and liquid interference. Okay. That cell migration from outgrowth. Then we discuss about dissociated cell culture. Dissociated cell culture also it's a disaggregated tissue. This is a disaggregated tissue. Okay. Then disaggregated tissue are the form monolayer. Okay. See this. This is monolayer and that form in solid liquid interference. Then we discuss about organotypic culture. Organotypic culture are different cell co-culture in this culture that are without matrix or with matrix that cell form the organic structure recreated. Okay, now we discuss about all these things. Then we lastly discuss in our cell culture application. We overvise this. The animal cell culture also used in cancer research, virology, then genetic engineering, tissue or organ replacement. This is a genetic engineering also include in tissue or organ replacement, stem cell therapy, then drug screening and toxic testing. We see this the vaccine production and 3D cell structure. That's 3D cell structure also include in the 3D human skin model that you use for the cosmetic industry. Okay. Now we discuss about the importance of cell culture in gene therapy. In this therapy, we already studied gene expression and gene theory, but now we studied in gene therapy importance. Gene therapy is more important because of that improving the quality of life. The changes in our body are showing the different disease. That disease are the fault of gene and mutation of gene and also the changes of some changes in the our genetic material and somatic cell and germ cell also. So what is gene therapy? Gene therapy is the cut the one part of DNA. That DNA are the study in vitro, and that DNA altered, yeah, that DNA are copy to progression in this the disease improving, yeah, solve the problem of this disease. The particular part of gene we cut and remove the this part, and that solve the problem for the help of gene therapy. See this, we underline this sentence: gene therapy involving insertion, removal, or alteration of therapeutic or working gene copy of cure a disease. Okay, then the cure of disease or defect or slow the progression of a disease. Okay, that help improving the quality of life. Another topic is another point is mm, genome map. That is more important. Human genome map. Okay, human genome map was the first major step toward new way of addressing human health and illness. Gene therapy hold great promise. However, the task of transforming genetic material into the cell remain an enormous technical challenge. Then the ex vivo cell cultivation and adaptation from the lab to a clinical relevant state. Okay. Now we discuss next point, that is the development of animal cell culture technology. This is a it's a advanced technique. We see this, the monogenetic disease are also that disease effect of single gene. Okay, that such as which disease are included there. That disease, the single gene defect is the showing cystic fibrosis, hemophilia, muscular therapeutical protein produced in the animal cell line. That time, the use of the solution solve the disease problem, the specific technology that advance to the importance of gene therapy. Okay, then we discuss another point, that's the gene therapy culture is more important. Identify the faulty gene. Okay, because of its help the solve the disease problem. Then another one is the help of gene isolation and generation of construct for correct expression. Okay, now we see this, the gene therapy importance in the delivery of genetic material in vivo or ex vivo in crucial to the success of gene therapy. Mostly the gene therapy are the not the easy to uh, insertion and deletion of gene, but the advanced technique using the gene therapy that easily solve the problem. Okay, then also it's in vivo therapy, the genetic material is introduced directly into the individual at a specific site. Okay, then ex vivo treatment, that the target cell are treated outside the patient body. Okay, we discuss here gene therapy importance, that cell are the expanded and transferred back to the individual at the specific site. Also, ex vivo technique involve gene therapy in the culture cell, which are expanded and subsequently transferred to the target tissue. We are discuss about the importance of cell, that cell of gene therapy. Gene therapy, what is the gene therapy? And we discuss about the importance of gene therapy. Okay, now we discuss about another point, that is the clinical correlation. Clinical correlation is more important. The one point is gene design. Gene design, it's a medication produced by the Shenzhen Simbono Gentech in China scientists. The first product designed by gene therapy, also called as Gendicin. Also, Gendicin is the used for gene therapy of cancer. That's the head and neck carcinoma treatment. Another one is the P53 gene and adenovirus are the formation of elimination and control tumor. Okay, the force suppressing gene P53 is recombinant adenovirus express protein P53, which lead to tumor control and elimination. Okay, last one is SBN cell is the cell line. Okay, that cell line was subcloned from the human embryonic kidney cell line, and it had been used for production of gendicin. We see this. It's a main key point is gendicin is the P53 gene plus adenovirus that's used the tumor control and elimination of tumor. Then next one is the gene therapy for the cancer, and another one medical breakthrough. What is the gendicin used properly? We know that the DNA small part is gene. That gene are the apply the adenovirus gene and P53 gene. They insert in the gene therapy in this human, and the adenovirus release the gene into the cell. Okay, that gene express the protein, and that protein are target showing the target cell. We apply for the uh, our that protein contain gene that also work on the target cell, and they form the receptor protein and secretory protein. See this. This is all about gendicin therapy, gene therapy. Okay. Now we discuss about organ culture. Organ culture, it is very simple concept. You understand it. It's a, see this is a diagram. This is an organ. We cut it and they cultured in media. This is all about all culture, organ culture. We see one by one that important point we discuss about here. First, whole organ from embryos that are used in initiate of organ culture in vitro. See this, in vitro we are cultured in vitro whole organ, also called as organ culture. Then the cell is the organ culture maintain their differentiated character, their functional activity, and also their architecture. Okay, all are more important to that maintain. That's why they are not progressing that culture. They are injured and not help to our extra work, another work we have done. It's not a help to that, because that character and function activity are more important. We record that and help to organ culture. Okay, organ culture is also useful for studying functional properties of the cell, that what we see this, the production of hormone, and for examine the effect of external agent such as drug and macro micro and macro molecules. Okay, the products on organ that are automatically placed apart in vivo. Okay, we see this all about organ culture. Now we move toward primary explant culture. Primary explant culture is the what is a primary explant culture? See this. We firstly understand what is the primary explant culture. The extra cellular matrix constituent that are collagen or plasma clot, and it can even happen to spontaneously added in tissue adherence. Another one is rise to cell migration from the periphery of the explant, and this culture called as primary explant. Primary explant are grow in outgrowth, also known as outgrowth. See this is outgrowth. Okay, firstly, primary explant is the proteolysis lead to production of single cell suspension. See this, and the suspension of single cell in this. Another one, the next diagram is see in this is the our mesenchymal cell surface cell culture here, and explant tissue pieces to here. This is all about primary explant culture. Now that what is used for primary explant culture is used to analyze the growth characteristic of cancer cell in comparison to normal counterpart. Okay, and especially with refers to alter growth pattern and cell morphology. That's why primary explant culture is more important, because of compared to cancer cell and normal counterpart. Now we are discuss about another point, that is the cell type and how its cell culture in our media. This is a series, all are cell, different shape and morphology showing here. Okay, we discuss about the basic cell culture, and we also discuss about cell type, characteristic and applications. Now we complete our fifth unit to in this lecture. We stop here. Okay.